Email from your wife probably while you're talking to her.
Ladies and gentlemen, let's begin the talk, and there are sandwiches at the back for our new arrivals. It's a great honor for me to introduce Professor Keasling. It turns out that we're friends, and we've developed a friendship over the last year or so, and I had the honor of meeting him for the first time through this very influential and $45 million proposal that was written between his office and a number of other organizations to take the drug to take this flower artemisinin and make these drugs for malaria, which I know he's going to talk about today. Not all of that money comes to the campus. Some of it goes to this important organization called One World Health that are doing more of what you might call the deployment and the rollout. The grant has a very citrus kind of flavor because it begins with very important scientific research, and then it narrows it down to this collaboration that essentially is helping other organizations roll out these very important social projects. Apart from the fact that Jay is a very gifted speaker and a really brilliant scientist, he's also very forward-looking, and he's the co-author of the Helios project, which, as you know, is a very large project up at the center of the LBL, and is looking at alternatives, especially solar alternatives, to fossil fuels. And there's another element of his work that's in the Helios project to replace fossil fuels with other biologically inspired materials in the manufacturing process. Please come in, have sandwiches and so forth. It really is indeed an honor. I'm sure that the talk is going to be fascinating and you'll be enjoying hearing many unusual things today. And I'd just like to begin with our traditional round of applause to welcome Professor Keasling.
Thanks, it's great to be here. So I want to motivate the talk by describing the disease we're trying to solve. It's malaria. Malaria is caused by Plasmodium. Plasmodium infects the red blood cell, consumes the hemoglobin, and reduces the carrying power, oxygen carrying power of the hemoglobin. It's transmitted by the Anopheles mosquito. Now, malaria affects between 300 and 500 million people at any one time. Every year, between 1 and 3 million people die. 90% of them are children under the age of 5. If that weren't bad enough, it's the fact that economists have noted that those countries most afflicted by the disease, shown in red here, their GDPs are reduced by half, because when you get the disease, it's very difficult to work. So it has tremendous impacts, bad impacts on the world. The drugs that have been most widely used and are widely available and affordable to people are those based on quinine. Quinine was extracted from the bark of that quinoa tree. There were wars fought over it, and it's been in use for hundreds of years. In fact, it's been used and overused and abused. So the result of that is that Plasmodium has grown largely resistant to the drug. So even though it's affordable to people, it's not effective. Now there is a drug that is effective. It's called artemisinin, and it comes from this plant, Artemisia annua, or sweet wormwood. Now this drug has a great history. It goes back to 168 BC, where there are writings of its use to treat hemorrhoids. And then in some later tombs, there are writings of its use to treat fevers, presumably fevers due to malaria. Now, the way this drug was gotten back then was by stripping the leaves off the plant and making a tea of them. So hot water will extract the drug from oil sacs that sit on the outside of the cells of the plant. The drug was largely forgotten until the Vietnam War. And there were problems getting a hold of quinine for the Chinese, and so they went back to their literature and revived this drug. And about 1972, the active ingredient, artemisinin, the structure was found. So it's got a great history. Now it's in a family of natural products. These are molecules that come from plants, animals, naturally. That's one of the largest family of natural products. In fact, there are 50,000 molecules in this family of natural products, and they include things like menthol, includes things like lycopene, the carotenoid that makes tomatoes red, beta-carotene that makes carrots yellow. It includes cholesterol in you and me. It also includes drugs like Taxol, the anti-cancer drug. Now, what most of these molecules have in common is that they're produced in very small quantities in the host in which they're produced. So they can be very expensive. In fact, Taxol is found in such minute quantities in the bark of the Pacific yew that it would take one 100-year-old tree to get enough Taxol to treat one cancer patient once. There aren't enough trees on the planet to supply all the treatments that would be needed every year. It turns out that you can get a precursor to this drug from pine needles and then derivatize it. And that's where it's gotten now. So I'm not going to show you a lot of chemistry, but I will show you a little bit. So these molecules come from isopentenyl pyrophosphate. It's a C5 precursor. It exists in every organism. It's a hydrocarbon. It's isomerized into dimethyl pyrophosphate, and those are condensed to form a C10, geranyl pyrophosphate, a C15, farnesyl pyrophosphate, and a C20, geranyl pyrophosphate. And then those are cyclized to form the precursors to those molecules I showed you on the previous page. So the family of cyclases that form these cyclic molecules are extremely diverse, extremely plastic. So you get the huge diversity, that 50,000 molecules, basically through this series of reactions that occur with those linear hydrocarbons. I'm going to talk today about one of these, artemisinin. It's a C15, so it comes from farnesyl pyrophosphate. And we're going to talk about this extensively and how it's made. Now, on the left, I show glycolysis and TCA cycle. So if you consume a sugar, it's going to go through the central metabolic reactions that are going to turn that into energy and precursors to make proteins, nucleotides, etc., to make biomass. Now, over on the left, the right side, I have the terpenes. Now, to get from the left side to the right side, you need a series of reactions. And in bacteria, these occur through a pathway called the DXP pathway. It's just a series of biochemical reactions that convert central metabolic precursors into the precursors for terpenes. And in you and me, the pathway that does that is the mevalonate pathway. Now, why that's important is because all of the statins, those anti-cholesterol drugs, target an enzyme in the mevalonate pathway. Okay, so a lot's known about this pathway because it's a multi-billion-dollar business to know a lot about that pathway. And that's going to be important in a minute. Now, let's go back to artemisinin. What's the problem with it? Cost is the problem. If you buy the drug as a traveler, you'll pay between $20 and $40 for a treatment. And this is a cure for the disease. That's too expensive for people in the developing world. So when the World Health Organization decided to recommend artemisinin as the drug of choice for treating malaria, they negotiated a price with GSK and Novartis of $2.25 to $2.40. This is a cure, but it's still an order of magnitude too expensive. What's more, because there are so many mosquitoes, you can be cured of malaria and then get it again and again and again. And this is the problem in coming up with a vaccine. Because if you could come up with a vaccine, then that would supposedly prevent you from getting malaria again. But here you've got this case when you've had malaria, you can get it and be cured of it and get it again. So there's a problem in coming up with a vaccine because having the disease doesn't keep, and being cured of it doesn't keep you from getting it again. Hence it would be hard to come up with a vaccine. So what's more, finally, is that when the World Health Organization negotiated this and decided to recommend it as the drug of choice, they didn't think about where they were going to get the artemisinin. Currently, they get artemisinin through small farms in Southeast Asia. And they extract the artemisinin from the plant using diesel fuel. So there's a little bit of a purity problem, and there's not nearly enough being produced. So ever since they recommended it as the drug of choice, the price has been rising, going the opposite direction that it needs to be. The goal for my lab in this project we've been working on is to reduce the cost of the drug by an order of magnitude by producing it in a microbe. and using renewable resources to produce it. Sugar, for instance. Now, if you're going to design an organism to do chemistry, you first got to know what chemistry you want to put into it. And that means you've got to go into the plant and you've got to find out what chemistry it's doing to arrive at your final product. Luckily, somebody did that for us, or at least started it. Bertia and colleagues reported the pathway that goes from amorphidine, which I'm going to talk a lot about. FPP is that C15 I showed you previously, to artemisinin. Then once you know what chemistry you want to put in a microbe, you've got to find the genes. And you can get them from wherever. They don't have to come necessarily from that plant. If you can find the chemistry elsewhere and it's easier to get, you go after it. Like, for instance, yeast. Can you get the chemistry inside yeast? Now, when we tried to get the first step in this pathway that takes FPP to amorphidine, from one of my colleagues who had reported on it in the literature, he said, no, I'm not giving you the gene. Because you're my competition. And in biology, we have this culture of sharing things once you've published it. So this was kind of a shock for us. So we got a gene that we didn't necessarily want, but we can get a hold of from tobacco that produces a molecule very similar to what we wanted. So we thought, oh, this is a good model. Turned out that it was a good model, but that the gene expressed extremely poorly in E. coli, and we got almost no product. In fact, absolutely miserable yield. So here's where we stand with this product. And that 1,000, that's a gram per liter, that's where we need to be. We actually have to be a little bit above that, but that's a good milestone for us. So this clearly wasn't going to cut it. And what's more, we couldn't get the gene. So if you can't get the gene, you can't buy the gene, why not steal it? And that's what we did. We stole the gene, and in stealing, we didn't actually go to his lab and take the DNA out of there. We took the sequence that he had in the literature, and we reproduced the gene. And the way you do this is you buy small pieces of DNA and you link them together to form a full-length gene. Now, there's a little bit of a problem, and that is that when you take a gene from a plant and you put it in a microbe, it's like taking an organ from somebody else and putting it in your body. The microbe rejects it, doesn't like it. So what you have to do is fool the microbe. You make the gene look like a microbe gene rather than a plant gene. And the way you do that is you recode it in the computer. And you just change the DNA sequence a little bit, and then the microbe doesn't necessarily recognize it as being foreign, and it expresses the gene at a much better level. And in fact, in doing that, and stealing the gene and making it better, we got about 142-fold improvement in the production. Now, these are the kinds of steps we want to make, because we still have 4 orders of magnitude there to go to get to the top. But it was clear to us that now this gene was pretty good that was making this precursor, but we really needed to work on the upstream pathway, producing this FPP. Now, you can do this by engineering this DXP pathway. Remember, I told you this is the pathway that microbes use, and we're doing this in E. coli, and it's got this pathway. So you could express some of the genes in this pathway and produce the enzymes at a higher level and eliminate any rate-limiting steps in the pathway. And we took 3 of these that were reported to be rate-limiting steps, and we expressed them in the microbe to increase the production. But we got very disappointing results, only a 3-fold improvement. Now, if we were in industry, they would say, oh, this is a great result. But looking at how far we had to go, we decided this just wasn't going to cut it for us. Now, we could go in and we could express every gene in the pathway. And that would have been fine, except the pathway wasn't completely known when we started this work. And that wasn't an option. Another problem is that when you express all of the genes in this pathway, sometimes you change the nature of those metabolites inside the cell. And some of them might be needed for cell growth. For instance, that DXP is needed for pyridoxine and thymine. These are 2 vitamins that the cell needs to grow on. Now, if we were producing an anti-cancer drug, I would just add these to the medium. The problem is that they're more valuable than the anti-malarial drug that we're making. So you can't very well add them to the medium if you're trying to produce a cheap anti-malarial drug. So we decided a different route, was just to bring in an entirely new pathway. And we chose the mevalonate pathway. It's known in humans and rats and mice and yeast. And so we brought the yeast pathway into E. coli. Now, I'm going to digress on some biology here because there might actually be a few biologists in the room. But I'll try to relate this more for a CITRIS audience, or what I suspect might be a CITRIS audience. So you take those genes, and these arrows are representations of the genes that we took. Some of them from E. coli, but most of them from yeast. And you string them together so that you can control the activities of those genes at various levels. And what we'd like to do is be able to turn on that entire pathway so that we've immediately got flow through the entire pathway to our final product. And we got great yields by doing that. We got almost 100-fold improvement by putting that non-native pathway in. And there were a couple of reasons. One is that we're starting with a metabolite that the cell makes so much of that, in fact, it tries to rid itself of the excess of that metabolite. So we're giving it an opportunity to get rid of it. What's more, this pathway comes from yeast. It doesn't occur in E. coli where we're doing this engineering. So it's foreign. It doesn't have any regulation. When we turn it on, it's on full blast. It's not throttled in any way like it would be in you and me, we hope, or in yeast. So it runs full blast and we get a lot of our product. Now, when you're doing this work, you try to balance the genes in those pathways because it's like putting pipe together. I'm a chemical engineer, so this is a good analogy for us. It's like putting pipe together. And each one of these genes is a different pipe. Well, if you're using 3-inch pipe and all of a sudden you've got a 3/4-inch pipe, the flow through the back end of that piping is going to be governed by that 3/4-inch pipe. And you can't have that because that's a bottleneck in your pathway. So you've got to make sure that they're all 3-inch pipe going all the way through your pathway. And one of the things we try to do then is to express each of those genes, our pipes, at the same level so we get the same flow through the pathway. But on occasion, you'll do things like you'll accumulate intermediates in that pathway. In one case, we accumulated IPP and DMAP. These are metabolic intermediates. Now, E. coli has these naturally in it, but when you produce them at a high level, they become extremely toxic to the cell and kill the cell. And what's happening is they inhibit the other pathway that E. coli has naturally. It's a regulated pathway. That pathway then can't produce pyridoxine and thymine, and therefore the cells can't grow. But if you balance the pathway properly so that your flow goes all the way through and these intermediates don't accumulate, then you can produce a lot of your final product. And so that was an important lesson for us. And there was another lesson in the top part of the pathway where we actually accumulated an intermediate in this pathway. In fact, it's an intermediate that's not so foreign to you and me, HMG-CoA. And I want to tell you a little story about this because it's important for human health, actually. This enzyme, HMG-CoA reductase, this is the enzyme that the statins target if you're taking them for a cholesterol problem. It's a regulated enzyme. And when you have a cholesterol problem, that enzyme is upregulated. So the doctor gives you a statin that targets that enzyme. It throttles the entire pathway down so that you don't produce a lot of cholesterol. Well, when we took this, the same thing occurs in yeast. When we took it from yeast, we took the regulatory portion off of this because we didn't want it to be throttled down at all. We wanted it to run full blast. And the problem is that when we put it in the cells, it actually became unstable. And so we had a bottleneck at this pathway, and this intermediate in that metabolic pathway accumulated and became toxic for the cells. I won't tell you what the toxicity is. It turns out it inhibits their ability to produce membranes. And so if the cells can't produce membranes, they'll pop. They won't grow. So the way to get around that is to deregulate that gene, open it up more, produce more of it, and you get more flux through that pathway. And we use some novel strategies, which I'll skip over, to increase the flux through the pathway. But all in all, we got about a 7-fold improvement in the production by balancing that pathway. So that was an important step in balancing the expression of this pathway. So we've come up now about 4 orders of magnitude in the production. But one of the things we're doing is we're growing these cells in a fermenter. And we're producing a hydrocarbon. And this hydrocarbon is just a little bit heavier than gasoline. And while we're producing this hydrocarbon, we're blowing in air continuously into the fermenter because E. coli needs oxygen. Otherwise, it produces acids and it pickles itself. And while we were doing this, all of this amorphidine, the precursor to artemisinin, was evaporating from the culture. So this is the culture with time. And the cells are gradually producing amorphidine until they run out of sugar. And once they run out of sugar, they quit producing it. And all the amorphidine then gets volatilized out of the fermenter and just goes away. It's like having a pan of gasoline out in the sun, right? It's just going to all evaporate very quickly. Now, there's a trick you can do. You can put oil in the reactor. Now, the oil doesn't just sit on top of the reactor. It gets mixed in. But it acts as a sink for the amorphidine. And so the amorphidine won't be blown out of the reactor. It's like adding gasoline to oil to a certain extent, because then it won't evaporate out of the pan. And what we noticed is that we were producing large quantities of our final product. We're producing about 0.5 gram a liter. So that put us almost near the top. So we're just about where we want to be. And that means that we've improved the production about 1 million-fold over where we started. Not bad. Now, what's left? We're right here at amorphidine. And we need to get to artemisinic acid. If we can produce artemisinic acid, then we can use standard synthetic chemistry that they use over there in Latimer Hall and produce any one of the derivatives. Artemether and arteether are great derivatives for children who have cerebral malaria. There's a blood-brain barrier, and this prevents a lot of drugs from going across from the blood into the brain. But if you have hydrophobic molecules like artemether and arteether, they'll cross that membrane and go into the brain and kill the plasmodium that's in the brain. I was speaking to an MD when I was at Michigan State giving a talk. She spends 6 months out of the year in Africa. She does autopsies on children who've died of malaria, primarily cerebral malaria. And she said the one thing that she'll often go into the hospital to treat patients as well. And she said when these children come in, it's as if they've been in a severe car accident and had head trauma. She said they're completely out of sorts. They don't know where they are. Most of them are comatose practically. They take the drug, and the next morning they wake up cured as if nothing had ever happened to them. It's amazing. It's a really amazing drug. The problem with these two drugs is that if they're given in large doses, they can be neurotoxic because they cross the blood-brain barrier. This artemisinin is probably the molecule of choice because the Army's been using it for years. Now, that drug doesn't have FDA approval, but the Army uses it on soldiers, so you don't have to have FDA approval to experiment with people in the Army. It's another reason not to go to the Army. And this is a great drug for intravenous use because it'll be soluble in water, can be given intravenously to children who need it. So that's where we need to be. But I want to talk about artemisinic acid because this is one of the most recent developments and actually will be published in Nature in about a month. So this is the pathway I showed you before that goes from amorphidine to artemisinin. It was outlined in that paper by Bertie and colleagues. And we knew that we wanted to get down here to artemisinic acid because then we can use chemistry to go the rest of the route. But that meant that we may need as many as 3 genes in that pathway. And one of those genes is called a P450. And P450s are genes that add oxygen to molecules. We have a lot of them in our liver. They are what detoxify drugs and other nasty molecules that we might ingest. And so they're very important. In plants, they're very important for producing molecules like Taxol, like artemisinin, because they add oxygens to them. But they're very tricky enzymes to work in microbes. They're very finicky. This is a picture of a typical cytochrome P450. It has at its center, you may be able to see it in yellow there, a heme group. And this is very similar to the heme and hemoglobin that carries oxygen. Well, hemoglobin carries oxygen. This protein puts oxygen into molecules. So it has a similar function. And it's very difficult to get that that iron center put into the enzyme. So we reasoned a while back that we were going to have another host. We decided, gosh, if this mevalonate pathway from yeast works so well in E. coli, maybe yeast itself would be a good host. And this would give us a way to screen for that enzyme. So this is just common baker's yeast. Now, here's how we go about finding those genes. Because we don't have them, nobody's published on them. There's nobody to steal the genes from, except the plant itself. But the plant probably has 30,000, 40,000 genes, and it isn't sequenced. How are we going to pull out the right one? Well, we decided we needed a yeast host that produced amorphidine, this product that I've been talking about all along, and that we would just screen. We would randomly insert genes into that yeast, and we'd look for the yeast that produced the alcohol, the next step down in that pathway. And that yeast would have the right gene in it. And then we take that gene and put it in E. coli. It's already producing this amorphidine, and it would produce our alcohol. So that's the trajectory we're going down. But first, we need this yeast that produces amorphidine. Now, I'm not going to talk a lot about the engineering because you've heard it for E. coli already, but I just want to make a few comments. And yeast produces a cholesterol-like molecule that's absolutely essential for its growth. It's called ergosterol. It looks almost like cholesterol, and it's essential to keep the membrane fluid. Now, if you add in the amorphidine synthase gene, this is the one we stole, into yeast, you get low production just like you did in E. coli. But we can start to engineer this pathway that produces the precursors to amorphidine by adding in various genes. And we add in more and more genes, and that works okay. What works the best was knocking out the ability to produce cholesterol, or giving the yeast cell just enough ergosterol that it could live, but not so much that it would turn all of the FPP into ergosterol. So we're stopping the flow here, or turning it down with a valve, and shunting the rest of the production into amorphidine. Now, right now, we're about an order of magnitude below E. coli in production, but that's good enough to go in and look for our gene of interest. And so we use that strain. Now, here's the path again. We're going to go into the plant and try to find that gene. Now, about that time, I hired a really smart postdoc who'd come from a terpene lab that works on these molecules. And he said, well, here's the molecule that we want. Here's artemisinin in Artemisia annua. And it turns out that lettuce, chicory, and sunflower are related to Artemisia annua, and they produce molecules that look a little bit like these molecules. Well, if it produces molecules that look a little bit like these, then those molecules, it may have enzymes that are very similar to the ones in Artemisia annua. And we have gene sequences for some of these enzymes. So we can use those gene sequences to try to narrow down the genes that we want to find in Artemisia annua. And maybe they're the same genes. And he was really good because he took those genes. This is a phylogenetic tree, and to you it might not mean anything, but basically what we're doing is we're taking the P450s from all of those plants, sunflower, chicory, lettuce. We're lining them up, and we're looking to see what's in common in all of those genes. And if you see what's in common, then it might be in common in Artemisia annua, and we can go for that gene from there. And that's what we did. And that 71AV1 is the gene we decided we needed to pull out of Artemisia annua. Now, about this time, there were a bunch of miracles that happened. I've never been much of a believer, but this changed my mind. The first miracle was the first gene he went after was the right one. Okay, that's out of like 30,000, 40,000 genes. Okay. The second miracle was when we put it into yeast, it was functional. It worked. The third miracle is that it didn't produce the alcohol it actually did all 3 steps in the pathway and went directly to the acid. And that data is shown here. This is a chromatogram. So we take an extract from the plant, we run it through a GC. The peak of interest comes out right here, the artemisinic acid. That's the same peak that comes out in yeast. When you don't express the P450, you don't get any of it. This is the mass spectrum, and what all you need to see is that they're identical. This is the molecule that comes out of the plant. The top is the molecule that comes out of yeast. 99% identical. It's there. It's exactly identical. So this enzyme now does all the steps in that pathway. So what we thought we were going after is just the first step, ended up being all of them. And that was great. Now, there was another miracle that happened, and that is that when we were looking for this artemisinic acid in the cultures, these yeast cultures, we couldn't find it. We looked inside the cells, couldn't find it. Looked in the medium, couldn't find it. Well, it turns out it was stuck on the outside of the cells. Now, what that means is that the cells produced it, and they secreted it outside the cells. But because yeast cultures tend to go to low pH when they're growing, this acid stuck to the outside of the cells and coated it. Well, what's important about this is this gives you a really easy way to purify the product. You grow the cells up, it produces the product, it secretes it on the outside of the cell and sticks there. So you centrifuge the cells down, you get rid of all of that medium, then you change the pH just slightly and it comes off almost in pure form. If the cells hadn't secreted it, you'd have to lyse the cells, and inside the cells are thousands of metabolites. And you'd have to purify it away from all those other metabolites. Well, the cell has essentially done the purification for us. It's got a transporter. It transports it outside the cell. And in that step, it purifies it. Okay. So, a bunch of miracles, and those are just outlined here. So, we've gotten the artemisinic acid. And now we're sticking it in E. coli. And right now, it's a race in the lab. I've got a group working on E. coli, and I've got a group working on yeast. And we're going to see who gets to the highest titers, and that's the bug that wins, and the one that goes commercial. Now I want to talk just a minute about the grant that we have and how we've set up this collaboration. The Gates Foundation grant involves 3 entities, as Paul mentioned. My lab, which does the gene discovery, constructs the initial strain. And then it goes to a company that was founded by 3 former postdocs from my lab called Amherst Biotechnologies. They're in Emeryville, and they do the optimization of the strain, because when it comes out of my lab, it won't be optimized to produce in the extremely high titers that you might need. And that optimization is not really great work for graduate students or postdocs to be doing. It's a little mind-numbing sometimes. And then there's the scale-up issues, getting it to work in a large-scale fermenter. Once it goes from there, it goes to the Institute for One World Health, a nonprofit pharmaceutical in San Francisco, where they do equivalency tests. Basically, they show that the molecule we've produced from yeast or E. coli is exactly the same as the one that comes out of the plant, if not pure, which is probably going to be the case. And then they find a large-scale manufacturer and get it out to people who need it. Now, another important aspect of this work was the intellectual property around the molecule and the process. We wanted to get the price as low as possible. And to do that, you want to make sure that nobody is taking royalties, who doesn't need the royalties. So we negotiated a deal with our Office of Technology Licensing here, who said they patented, or we've patented, all of the strains that we've created. And they said they'll license them to this group, to Amherst Biotechnologies and the Institute for One World Health, as long as they agree to produce the drug at cost for people in the developing world, not take any profit from it. And the university won't take any royalties. So there are no added costs onto the drug from any of the people who are working on this grant. Now, clearly, to keep people motivated in pharmaceutical companies, they're going to have to make a little profit on this. but that's fine with us. The main ingredient, artemisinin, we will have reduced its cost from about $1 a gram to about 7 cents a gram. And that's the levels we're looking for right now. And they'll still be able to make a little profit, but it'll reduce the cost of the drug, hopefully by an order of magnitude, once it gets to people in the developing world. Okay, so what I've told you about today is that we've engineered E. coli to produce amorphidine, a precursor to artemisinin, at about 0.5 gram per liter. We would like to get to 25 grams per liter, and we're getting very close to that. We're much higher titers than what I reported here. The E. coli can actually express this P450, although not as well as yeast. The yeast, we've got production levels of amorphidine at about an order of magnitude less than E. coli, but still quite high, and it produces this artemisinic acid and secretes it out of the cells in pure form. So as I said, right now it's a horse race between the two organisms, and frankly, I hope they both win. These are some of the people in my lab and at Amherst Biotechnologies who did the work. The work was funded by the National Science Foundation and the UC Discovery Grant early on, and since then it's been funded by the Bill and Melinda Gates Foundation. And I just want to put one plug in for a meeting that's coming up, Synthetic Biology 2.0. It's going to be held in Berkeley, May 20th through the 22nd. It's going to be an exciting meeting where we're going to talk about, I won't talk about this application, but other people will be talking about applications like that, as well as the foundational technologies that allow us to build organisms like the one that produces artemisinic acid. So sign up early, because we've got about 250 applicants right now for 350 slots. And we expect that it will be oversubscribed. It's been a pleasure to talk to you. I'd be happy to answer any number of questions. And I thank you for your time and attention.
Well, I knew that would be a great talk, and that certainly was fantastic. Thank you very much.
Sure.
Just really wonderful. So we have the floor for about 10 minutes. Jump in and ask some questions. It would be great to hear. So, carry on.
So you've talked several times about turning the production of some chemical up or down. How do you actually do that, since obviously there's not a little knob in there? Right.
There are little knobs in there. We really just don't turn quite the same way as those on the wall. We take a gene and we place it in the cell under the control of what's called a promoter. A promoter is a sequence of DNA that turns that gene on or off. And you can often control those promoters by adding a chemical to the medium. The chemical goes into the cell, and then it interacts with the protein that binds to the promoter and turns that gene on or off. Now, many of those are digital switches, but we have a few that are analog, so that we can actually add gradually more and more chemical to the medium and turn the genes on to higher and higher levels. That, when you turn the gene on to a higher level, that produces more protein. When you produce more protein, you get more flux through the pathway. So there are a lot of steps between that dial and actually turning flow up through that pathway. But yeah, we do have lots of switches. Now, the big problem for us is we put 12 genes in there. So in theory, we should need 12 switches. But what we try to do is put the genes in series so that we can essentially turn several genes on. In a couple of cases, you saw 5 and 7 genes on with one switch. And this gives you the ability to turn on an entire pathway, or turn it down, or gradually move it around.
Yeah.
Yeah, I heard you talk about 1.5 years ago, and you explained in more detail something you skipped over and alluded to now, that is the dials are able to actually be introduced by controlling the lifetime of the messenger RNA.
Yes.
That's a fascinating result and a great contribution, I think. And at the time you were saying that you were hoping you could swap this idea, license this technology to people that would provide you excess fermenter space or fermented capacity. did that go anywhere? And if not, how do you plan on doing the fermentation?
Right. So just to give you an idea, so to get let's say 500 to 700 tons of artemisinin or some derivative of artemisinin that we'll need annually, that means that we'll have to have about a 250,000 liter fermenter running 24/7, 365. I mean, it's just round the clock. So, but as it turns out, there is excess fermentation capacity out there. It's available in companies. I was in Austria a few weeks ago visiting Sandoz, and they have 7 of these things strung together, 7 250,000 liter fermenters. I mean, they're 4 stories tall. They bring in glucose in on freight cars. They're consuming about 12 tons of glucose an hour in the plant. There's fermentation capacity in this country. Now, the trick is going to be getting fermentation capacity really inexpensively donated, because that would really reduce the cost significantly. And for the drug companies, they get the PR from it. And you may not think that that would convince them, but if they've got excess fermentation capacity, that PR can go a long way, especially when they're selling their other drugs at high profit.
Andrew, then.
How much processing does it take after you get done with it? I mean, so you put it in the fermenter, then what happens?
Yeah, so then you've got to purify it out. And that usually is an extraction step. So it's going to be in water. If it's artemisinic acid, you've got to pull it out of the water in some way. And then it's got to go through a chromatography to purify it. These aren't unusual steps. They aren't difficult steps. What's important is that we have the biologists and the bioengineers who are doing the engineering of the bugs starting to talk to people who are going to be running the process, because those process decisions can affect how you engineer the microbe and vice versa. Like this little finding of, which wasn't little, of yeast being able to secrete the drug, that makes a huge difference in the pipeline after the fermentation. It simplifies the purification a lot. But so you need to take all those into consideration. So all of these people have to work hand in hand on developing the process.
Along that line, then, does the secretion help then the yeast essentially make a continuous process instead of a batch process?
Yeah.
And take the product out so it produces more.
Right. That's a great suggestion. Could we, in fact, take a stream off, centrifuge the cells down continuously, pull the product off, and then recycle the cells back into the tank? And the benefit to that is that you don't have to make more cells, right? So how you get this cheap is the cells grow on the carbon, on the glucose that you put in. Not all of that glucose is going to go to your product. Some of it's going to go to making cells. And in some ways, that's a waste. And if you can minimize what goes to the cells and maximize what goes to your product, then you've made a cheaper product. So by doing this continuously and not having to make more cells, you then could have a higher conversion to your product. So I think that's a great suggestion, and we hope so. The other important implication of the yeast being able to secrete this is artemisinic acid is most likely toxic to the yeast. This pump keeps it out of the inside of the cell where it's going to be toxic.
The second question then comes back to the delivery of the material.
Yeah.
Is that orally or
right? So most of the versions right now are oral. You can get tablets. I should mention the tablets. So you may have seen an article in the New York Times. Well, you may not have been looking for it, but I see everything that says artemisinin. And the World Health Organization was discouraging the use of artemisinin, not because it wasn't effective, but because some companies were selling it as a monotherapy. It's now sold as tablets by GSK and Novartis as a combination therapy with another drug. It's just like the HIV drugs that are being recommended right now. They're sold as combination therapies because you have less of a chance of the organism, either HIV or Plasmodium, developing resistance if 2 drugs hit it than if 1 hits it. So these pills are sold as combination therapies. But there are a lot of companies out there, rogue companies, that are selling it as a monotherapy. That's one problem. The second problem is there are a lot of companies selling artemisinin therapies that don't have enough artemisinin in them. And the problem, and they're selling them for cheap, so people are buying them. It may have 1/10 of the dose that's required to kill Plasmodium. So it doesn't stop the disease, but in fact it encourages more resistance. And then there are the third group of companies which are selling artemisinin therapies that don't have any artemisinin in them. And this is a huge problem.
So does the plant itself kill the malaria in the mosquitoes?
You mean like if the mosquito...
Also, like in areas where the plant grows, are there fewer mosquitoes that carry malaria? Is there a way to use this just to prevent it at the source, I guess is what my question is.
Well, so I think that's not the case because the plant grows well in Vietnam, and Vietnam is one of the areas where it's the worst. But let me answer a question that's similar to that you didn't really ask, and that is, could farmers plant the plant and get more artemisinin? And that's being explored. And I have to put a plug in for the Bill and Melinda Gates Foundation because these people are funding like 95% of all the research on artemisinin and all of the cures, and so it's not just they're funding our grant, they're funding other grants to encourage farmers to plant Artemisia annua so that there's a potential supply there. They're funding grants to discover drugs that look like artemisinin but that could be synthesized simply in the laboratory. And I think they're taking the approach of we're going to try all of these because there's not going to be one single solution to this huge problem. It's going to take many solutions. So they've really done the right thing. But getting back to that, there are farmers planting this crop. The problem is that production is affected by the weather. They're not making much money. And they can't really process the plant themselves. It has to go to a pharmaceutical manufacturing facility to be formulated. So we'll see how that works out.
Gary, let's make this the last question.
Oh,
Jump
gosh.
in. No, it's okay, because we're getting close to the end.
This is a project, Jay, which is obviously of enormous potential impact on the world and a beautiful piece of work. And I'm sure that either you or others you'll move on to something else. I'm trying to think of how one has the vision and the how do you pick a project like this? What do you see in other areas where there's a need and there's a potential way of getting to it? Because the way that you've gone about this is not just having the impact in that area, but it's sort of a model for choosing something that's of enormous potential social benefit, uses science in the right way, develops tools. I mean, all the stuff that's come out of this there may be a methodology there, but the key part of the methodology is picking the things to work on. So Paul's working on energy. I hope it has a similar impact. I'm looking for a way. You must have stopped to think, gosh, what else could we work on that you could do something like this with. And so from the CITRIS perspective, those are the kind of things that we want to really incubate.
Sure.
So your insight there on how do you
I wish there were a formula because I'd probably be really rich if there were a formula.
You're really rich anyhow. I want to hear. Have you thought about other areas?
So I have to say that some of it's timing, some of it's luck.
Of course.
A lot of it's persistence. I think there are a lot of good untapped problems like this. The developing world is full of these kinds of projects. But I do want to say that, part of it is being at the right place at the right time. We happen to be working on this metabolic pathway. when this drug was recommended by the World Health Organization. It happened to be a shortage. This was something we could synthesize. But it took a lot of money also to make this happen, and a lot of people. We probably put in a couple hundred person years into this project.
I think that that's the kind of thing that, just because this is a CITRIS seminar, that's the kind of thing we ought to be looking to incubate, fund, launch, and get a lot of wood behind those arrows. And so insights like the one that you just shared, I think are very, very useful to Paul and to Shankar and myself.
You know, let me just put a plug in for, since this is CITRIS, for cheap sensors for these kinds of things. A lot of these drugs are overused because there's no dipstick for malaria. We need a cheap, almost like pH paper for malaria. You prick your finger, you put it on a piece of paper, you got it or you don't.
Sounds good. Okay, who wants to work on that? Oh, you do. Great. No, you keep on working on too many things.
If the right dose kills it in a body overnight, then is there any way to make the litmus test out of your drug? I mean, not to say that there's enough to go around right now, but once the production lines are in place, it seems that could very well be. Potentially
there could be something around that. But I think, and I think for most of these developing world diseases, this would be a huge gain for people, because diagnosing something is a nightmare over there. They can't do it, and so they often give children particularly anti-malarial drugs because they just don't know.
Yeah, they want to be safe.
Yeah, and I would too, in my case. So.
Folks, this is a great seminar. Let's thank Jay once again for